in our compound light microscope, the smallest possible distance that can be resolved is 0.2 micrometers. So if we do the math, if we compute everything, okay, the minimum distance that can be resolved is 0.2 micrometers. Meaning to say, we have minimized our numerator as small as possible, and we have maximized our denominator as big as possible. So the question is, how can we minimize the numerator, 0.61 lambda? Take note that lambda is the wavelength of light, and 0.61 is a constant, so we cannot do anything about the constant. But which light color has the smallest wavelength? The answer is violet. And that's the reason why in some of your microscopy activities before, if you can remember, there is a blue filter that we insert through the lenses so that the image will appear blue. And the reason for that is we are really trying to use a shorter wavelength for the light, thus minimizing our numerator here, 0.61 lambda. And how do we maximize the denominator as big as possible? Well, we simply have to use a medium that has the same N, what is N again? Refractive index all throughout as the glass of the objective that we are using. So let me explain that later on a bit more clearly. So here we have a series of images with different sizes. So these are molecules from atoms actually to the size of a frog egg. Okay? And notice here our conversion factors for length. So since the smallest possible distance that can be resolved through a light microscope is 0.2 micrometers, we can therefore say that we are only able to view the specimens highlighted in yellow through the microscope. Meaning to say we cannot see the HIV particle or the HIV virus under the microscope because its diameter is 100 nanometers. That's less than 0.2 micrometers. But we may still see the cilia or cilium because a cilium has a diameter of 250 nanometers or 0.25 micrometers. So let's pause for a moment. Maybe you want to answer this, and in our future video teleconference, I will be asking several people, why do cells need to be so small? So, let's talk about visibility now. In the macro world, the normal world that we are seeing, we see things because light is reflected by those things back into our eyes. But in the microscopic world, this is the mechanism of seeing things is a bit different. It's not through reflection, but instead it's through the passing of light through them. So here's an experiment or a thought experiment. If you put a glass ball into an empty glass, you would be perfectly seeing the ball with your eyes. And why is that? Because the glass ball's refractive index is different from the air that's surrounding it. Meaning to say, as light passes through the glass ball and into the air surrounding the ball, it actually bends a bit because their refractive indices are different. Now, what if we put a glass ball, same glass ball, in a glass of water? It might be more challenging now to see the ball in the water because the refractive indices of the ball and the water are a little bit closer to each other now. So it will be a bit more difficult to spot the glass ball. But what if we put the glass ball in an oil pool inside a glass, like the immersion oil. The thing is, the refractive indices of the glass ball and the immersion oil are equal. They are the same. Therefore, the ball literally disappears from our view and we don't see the ball anymore in the glass of immersion oil. So it is now invisible. So this is how we see specimens microscopically, because light passes through them and light sort of bends as it passes through them because of the differences in the refractive indices. So here's a reason why we need to use immersion oil in order to use the immersion oil objective. Here in the left, we can see the absence of the immersion oil. And notice the light rays in green as they just bounce back through the cover slip or bend through the air, the gap between the specimen cover slip and the objective lens. And the ultimate effect is a little bit less light reaches our eyes here in the left. But on the right side, when we use the immersion oil practically has the same refractive index as both the cover slip and the objective lens. Therefore, as light passes through the objective lens, more light may do so, because there is now less bouncing back or bending of light, because the oil allows the whole thing here to be as if it's made of the same material, because their refractive indices are equal. So there's more light and we can see more clearly the specimen. And lastly, it is important for our specimens to be thin enough so that light will pass through them. But cells in their natural state are usually colorless, okay, like jelly without color. So it's important to stain them. The staining of these specimens will allow an increase in the contrast in the image that we see under the microscope. So this is why we stain our specimens when we view them under bright field light microscopy.